Okay, Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Inshallah, we'll start the questions now. I think in this chapter, if you remember, we started with the banana plants that are grown, wild bananas having seeds, and those bananas that are grown from the graft or are suckers having roots, asexual reproduction and sexual reproduction in plants. And then if you remember different types of cells that are involved in sexual reproduction, we mentioned pollen grains and also ovary ovules and seed formation, fertilization, seed formation, and then germination of seed. Okay, so all these questions are focused on the same major learning objectives that we were focusing on. So let's go to question 1. So I would like to have some volunteer, those who would like to read the question and they would like to answer as well. So first 2 question, who is going to answer and read it as well? So you can just raise your hand. Yes, Beta Yusra, you would like to start? Go ahead, yes. Okay, yes, Yusra is fine, yeah, go ahead.
Yes. Can you hear me?
Yeah, we can hear you.
Yes. A sex cell, it can be male or female.
Okay, start from the match each of these words, read it please carefully. So each one is focused. I know you can see the slide, but be focused. What question is asking about? Okay.
Okay. Question number 1. Match
Okay.
each of these words with its definition.
Uh-huh.
A sex cell, which it can be male or female.
So you have to
Gametes.
take it from the bank. Yeah, the word bank, they have given you the options.
I think it's gamete.
Yes, gamete has got both. Very good. That is correct. Yeah. So male and female. Would you like to name them? Because we discussed that, the exact names of these gametes.
For the male, it is the sperm, and for the female, it's the eggs.
Or ovum. Okay. Yes.
Should also do B.
For the plants, male gametes and female gametes. What are the names of the plants, male gametes and female gametes?
Pollen grain for the male and ovule for the female.
Yes, that's correct. Okay. Yeah, go ahead for B as well. Yes.
B. A cell formed by the fusion of the nuclei of two gametes. Zygote.
Yeah, this is a cell which is formed by the fusion of nuclei of the two gametes. Yeah, exactly. So that's called the zygote, right? And what is the process called? The process of fusion. If you pick it, sorry.
Meiosis.
The process of fusion of male and female gametes is called? A zygote is formed. Yeah, that's true. You answered the question. I think this question is asked later. You could see the answer is here. Okay, we can find it later. Okay, no problem. Let's go. Someone else would like to read questions C and D. Who is going to answer C and D? Read the question carefully and then pick up the word that is matching this description. Anyone would like to? Other than Yusra, Yusra has already done 2 questions. Yes, go ahead. Somebody would like to raise hand? Yes. So you just can read it, okay? A type of cell division used in growth and asexual reproduction, which produce new cells, genetically identical to the parent cells.
Sir, at that time when you were asking me the process, I couldn't hear your voice clearly, but I think it's fertilization, the process
Very
that you
good.
were asking.
Yes, very nice. Yeah, that's true. That is called fertilization. The process is called fertilization, and the cell which is formed is called zygote. Yes, the fusion of two half genetic material from one parent and or half from another. So fusion is taking place, and that is called fertilization. And the single cell which is now formed and this is now diploid is having is called fertilization and the cell is called zygote. Okay, so what about C? You would like to read, Yusra, C as well?
Yes. C. A type of cell division used in growth and asexual reproduction, which produces new cells genetically identical to the parent cell.
Yes.
It is mitosis.
Why not meiosis?
Because mitosis is the cell division in which the DNA is replicated, but in the meiosis, it is the division of the chromosomes
Yeah,
from diploid to
yeah,
haploid.
exactly. Very good. Meiosis only in case of sex cell formation, while mitosis generally for just exactly the same cells are produced. In meiosis, what happens basically when the cells are dividing? Of course, initially this sex cells, either sperm or ovum or egg or pollen grains, they are basically coming from the parent cell, right? Specific organ, the sexual organ that are producing sex cells, and the anthers that are producing pollen grains. So they are dividing. The cell are divided, of course, but they are having half goes in one cell and half, like you have two cells, meiosis 1 and 2. We will study that in more detail, I think, later on. But yes, so it is dividing. The chromosomes are also dividing into half. So if you have two sex cells, so one sperm has got 23, another cell has got 23. Okay, so this is how the division will take place. But in mitosis, yes, you rightly mentioned that the chromosome number will remain the same, and that is why we call it asexual reproduction, right? So in plants, especially that are grown from the same by asexual method, you would see the same number of chromosomes, same genetic material as well. Okay, what about D? Who's going to read D? Aisha beta?
The transfer of pollen from an anther to a stigma is pollination.
That is called pollination, yes. Would you like to tell us what are different types of pollination and different modes of pollination? I just want to recap some of the things that are needed. You might be asked question related to that as well, so that you can easily integrate those answers. Okay, so what are different types of pollination, first of all, and then what are different modes of pollination? How pollination will take place? These are two questions. Who is going to answer? Aisha, you would like to answer?
No.
Anybody else? Anyone else? Nabil?
There are two types of pollination: wind pollinated flowers, wind pollination, and insect
These are the
pollination.
means of pollination. I'm talking about the types of pollination. Types. Right? Types you mentioned. Yes, Hamza?
Sir, cross-pollination and self-pollination.
That's true. Yeah, these are two different major types: self-pollination, and then you have subtypes. Always, it's in the same flower in the same plant, or the flower is on a different plant. I don't know. They didn't mention in your book, but yeah, there are much details, fine details. You can go through it. But yes, two types are self-pollination and cross-pollination. Now, Nabil, you were talking about the means of pollination. Go ahead, please, with this explanation: wind, water, whatever you were saying. Please go ahead. Yes, Nabil.
So there are two types of pollination: wind pollination and insect pollination. In wind pollination, there are small or no petals at all. Should I explain them?
Oh, it's okay. It's okay. Yeah, yeah. Go ahead.
In wind pollination, there are small or no petals at all. They have no scent. They have no nectaries. They have stigma outside the flowers. They have pollen grains made in large quantities. They have light pollen grains, and they have anthers dangling outside flowers where they can catch a wind.
Yeah, that's true. Yeah. So in case of wind and insects are bird. Yeah, very good. Yeah, that's true. You already added much information, mashallah. Yes, excellent. So this what whenever you ask a question, when it's a pollination, it should come to your mind. What kind of pollination is this? Cross pollination, self pollination, and what are different means? Okay, who is going to read E and F? What is E? Yes, Nabil. E and F. Yeah, these two options. Okay, Asha.
An ovule after fertilization is a zygote.
An ovule after fertilization. Okay, but you already picked the zygote, right? For this one, a cell which is formed by the fusion of the nuclei is called zygote. So do you think if there is a, you could definitely use it multiple times if it's really true, but in ovule after fertilization, anything else here you think? What happens? You know, what is the next stage? What will be formed after male and female gamete are fused, fertilization has taken place, zygote is formed. So what is moving forward? What will be the next stage in case of plants?
Sir, embryo?
Embryo, or you could say seed. Yes, seed. So they have mentioned seed here, right? So it's going to be seed formation. So remember that when pollen grains or nuclei of the pollen grains are fused with the nuclei of the ovules, so you have fusion, right? This fusion is fertilization, and this fertilization is called zygote, and this zygote is growing into seed, and also the ovary will grow to become fruit and all the stuff, right? So the best answer here is seed. You have used zygote. Zygote is a fusion. This is very accurate description of zygote, right? And what happens after an ovule then? Okay, what about F, Nabil or Aisha? Who is going to answer this?
The fusion of the nuclei of two gametes is fertilization.
Yeah, you already mentioned here. Yes, exactly. The process, this process is called fertilization. Very good, excellent. Right. Okay, so let's move forward to question number 2. So who is going to read this question number 2?
Sir, may I?
Yeah, okay, go ahead.
Construct a two-column table with the headings sexual and
I think he's disconnected, right? Something.
hello, sir. Can you hear me?
Yeah, we can hear you now. Yes, Nabil, go ahead.
Construct a two-column table with the headings sexual and sexual reproduction. Write each of these statements in the correct column.
Yeah,
First,
so you have to
only
draw 2 columns, one asexual reproduction and sexual reproduction. So now, Nabil, what do you think of the first 2 options?
first is asexual and then sexual.
First part is asexual reproduction. First, only one parent is involved.
Yes, sir.
Yeah, that is asexual. Very good. But in case of sexual, 2 parents are involved, right? Because this is a fusion of both male and female cells, right? Gametes, right? So male and female gamete fusion is only possible in sexual reproduction, not in asexual reproduction. Okay, 1 or 2 parents are involved.
So sexual reproduction.
So what are those parents that are called, that are one parent producing both gametes, male and female? What do we call that? If this is one parent is involved, this is asexual, but they say it might be that 1 or 2 parents are involved. So 2 parents, of course, this is for sexual reproduction. But what about 1? Wouldn't it be asexual reproduction?
Yes, sir, it would be asexual.
But why didn't they mention? But then where would you put it? You would put it under column asexual or sexual reproduction?
Say asexual.
Oh, okay. So who is going to comment on that? Is it correct? Do you think we should put it under asexual reproduction? If you want to put in our asexual reproduction, probably a part of maybe fitting because 1 parent is involved, but this 2 parents, then it's maybe wrong, right? We cannot put it. Anybody would like to comment on that? We discussed this in the chapter. So those organisms having both male and female gametes are called hermaphrodites. Remember hermaphrodites, like earthworms? So it's 1 parent can have both. So that's why they say 1 or 2 parents. If one parent is a hermaphrodite, so in that case, it has having both. Plants, flower is also having the same male and female in 1 organ, in 1 part, right? It's not like human. So that's why 1 or 2 parents. That's why so it would go under sexual reproduction, not asexual reproduction. Because if you put it under asexual reproduction, 1 parent, they didn't mention, but they connected 1 with 2 or. So I mean, these options both are possible. There's only possible in case of sexual reproduction. And those organisms having hermaphrodite, you mentioned those organisms having both gametes, male and female, in one body in the same organism is called hermaphrodite, both characteristic. Okay, we discussed, I think, skull or earthworms, the most common, and also plants. Okay, right. Who is going to? Is it clear, Nabil? Because first we were thinking we should put it under asexual. Would it put it now under sexual reproduction? Is it clear to you? Okay, Nabil is disconnected. Okay, go ahead. Who is going to read next one? Nabil, is it clear?
Yes, sir.
Okay, go ahead. Who is going to read next part?
May I continue?
Yeah, okay.
Involved gametes involve fertilization.
Uh-huh.
Sir, may I answer?
Yeah, go ahead. Yes.
Therefore, involving gametes, it's sexual reproduction, and for fertilization, also it's sexual reproduction.
Yeah, because always either it's a plant cell or animal cells. And when there is involvement of gametes mean male and female, and there is involvement of fusion, which is fertilization. So definitely sexual reproduction. You cannot think about asexual reproduction. So when you talk about asexual, you could think about tubers of potatoes, or you can think about banana plant that are grown these days from the suckers, or grafting, when you just cut a branch of a plant and then you plant it somewhere in the soil so it will grow into a new plant. This is asexual because there is no genetic diversity. This is extremely important. Okay, zygote is formed. Yes, who is going to answer? Somebody else would give a try. Yusra?
Zygote formed is
Yeah.
sexual reproduction.
Okay, what about the next part? You would like to cover that next two?
Yes, all offspring genetically identical. This is asexual reproduction.
Very good. This is asexual, genetically identical. Why they are genetically identical?
Because this is asexual reproduction, which means mitosis process is happening in which the DNA is being replicated.
Yeah, there's no division of chromosomes. Chromosomes are not divided and they are not cross-linking and no genetic transfer. It's almost the same, dividing by mitosis. And one of the best option we discuss in your book are the banana plants and the potato tubers. So you will put it under asexual reproduction. Oh, excellent. What about next?
Can I do this?
Yeah, please go ahead. Yes.
Genetic variation among offspring. This is sexual reproduction.
Yeah, genetic variation among the offspring. Yes, true, because there's a meiosis and this will definitely take place. And yes, okay. What about next question? Who is going to do the next part? Somebody would give a try to this part. It's a little bit testing your analytical abilities. They want you to focus on experimental setup. This is the whole setup. You should not miss any of the information. First of all, how many tubes in sequence A to E, right? And then what is there inside? Inside are seeds, okay? And there's cotton, okay? And this is moist cotton, and seeds are the same. So all seeds and cotton is there. The only thing, this is moist, this is dry, okay? So this way you have to analyze the whole experiment. You try to understand what is common, what is uncommon in these tubes, first of all, before you answer any question. So whenever, don't jump. That's why I ask my student, don't jump to conclusion. That is using your analytical abilities. How do you analyze information? How do you group information? It's not straightforward question that, okay, which one is doing what? Yes, no, which option is correct, right? So that is kind of, you could say, short answer question as well, where you have to write a little bit about the data that they have given you and the information. So who is going to give a try? This is a very, very good question. Not necessarily to answer is fine. If you don't want to answer, this is absolutely okay. Who is going to read it? Yeah, Saad, go ahead, Saad.
A student investigated the conditions needed for the germination of mustard seeds. The diagram below shows the apparatus at the start of his experiment. Tubes A to D were placed in the laboratory at room temperature. Tube E was placed in a freezer at -4 degrees Celsius.
Okay, so first, what exactly this experiment is about?
About seeds.
Yeah, so seed germination. Okay, this is the purpose of experiment, seed germination. And now you can also pick on other information. What are other information they are giving you immediately before looking at the tube? So what are other information? You try to group information, organize your information, then you cannot make mistakes. You can directly go to the answer immediately. If you spend little time on understanding the scenario, and then definitely you could answer very easily. So one thing, yes, I agree with you, Saad, mashallah, that this experiment is about germination of seed. Okay, what are other information? I want some student to tell me, what do you get from, what are the other information they have given you in this paragraph? Yes, Yusra?
All the tubes from A to D were kept
they are
at room temperature
Yeah, just
except
one
for
second.
tube.
Nabil, Yusra is answering, so we'll give you a chance. Please go ahead, Yusra.
All the tubes from A to D were placed at room temperature except for tube E, which was placed at -4 degrees.
Wonderful. This is another very, very important information, right? So from A to D, they have given you the same thing, okay? But E is at -4 degrees Celsius. So that is very, very important to see. Another information, okay? And now you would look at the experimental setup before they ask you what is going on, look at these labels, A, B, C, D, and what is over there. As I mentioned, C's are there, dry cotton. In here is boiled water and oil on the top of it. Okay, the seed is inside the water, fully sinking in water. And here is little different, right? They are saying what is different here. So how this is totally different, or which aspect is testing more if you compare with other tubes? Yes, Nabil? How this D is different? If you'll focus on other tube, yes, Aisha?
It's not letting in the sunlight.
Wonderful. Yes, very good. This is
Can you
another,
repeat?
this is very important. Yes, the light is, because it's a black card. The rest of the information are the same. It is called moist cotton, which is here also moist cotton, okay? But it has a very special card which is placed on this tube not to allow light. And this, as you mentioned here, all the same moist. So no problem with the water contents, and no problem with the seed, but yes, temperature is different. Now you could easily understand what is different, what is common. What is common? All of them have cotton and seeds. Okay, this is common for all. So what is uncommon? Some are dry cotton, some are moist, some are totally submerged in water. Okay, and there's another difference in temperature, that one is at room temperature, another is at the -4 degrees Celsius. Yes, Nabi, you want to add something, or you have comment?
No, sir.
Okay, so now let's see to the what they are asking about. So Nabil, go ahead. Part one.
Which one of these factors should the student have kept same for all the tubes? Choose one from the list. Age of seeds, amount of water, temperature.
Yes. Yes, Nabil, you can answer as well if you know the answer.
Sir, I think that it's age of seeds. I think.
Why not temperature? What do you think? Because if the temperature would be constant, wouldn't it affect the germination, do you think?
Yes, sir, it will, as it is a factor of it.
So what makes you say that? That's true, that the answer is correct. Age of the seed is very, but how? I mean, why you didn't pick temperature? Why did you pick age of seed? Is there any special explanation, reason for that? Anybody would like to comment? So this is, look at this amount of water, temperature. First of all, look at this one, amount of water. If it is dry or moist, would definitely affect, but not up to that extent. But what if you keep all the thing constant and the age of the seed is not constant? Yes, age of the seed is extremely important, because seeds that are old enough, they might be damaged because of the disease, or maybe the enzymes are already non-functional. Even if you you plant it, is not going to grow. So age of seed is very, very important. Even if you kept the temperature constant, all the other thing constant, but the age of seed is different, you won't be able, it is possible that the quality of the is, because you want to see germination of seed. What is the objective? The objective is to see how seed is germinating and what are the conditions needed. So you have to look at the quality of the seed. If some seeds are damaged, they are not going to grow. So you won't be see, you might have put here for example, old seed, moist. It has got all the good environment, room temperature, moisture, light, everything is there, but it did not grow. It is possible that the seed quality was not good. Seed might be damaged, eaten up by the pests or eaten up by the insects, right? So if the cotyledon is damaged, it's not going to work. That's important. So that is why you have to keep the age of the seed constant, so that you could understand that the quality, because this is the indicator, right? This is the indicator if it grows or not. If this is faulty, the whole experiment is faulty. It's not going to give you a good data. Do you agree with me? Does it make sense? What do you think? Do you have any other opinion? Okay, so yes, Nabil, got it. Okay, what about option 2? Who is going to focus on option 2? After 3 days, the seeds and tube B and D had germinated. Germination taken place in B, which is this, and also in D. D is also taking place, B and D. So the seeds and all other tubes had not germinated. So using this result to deduce the condition, or to conclude the condition needed for germination of seed. So they have given you now experiment that B and D were, the seed quality was the same. So you are a bit sure that each tube has got good quality seeds, okay? So B and D is germinating, E is not germinating, C is not also germinating, A is not germinating. Why? What comes to your mind? What makes special about B and D? And you can use your common sense as well. When we are planting seeds, you remember, you have seen farmers, they plant seeds in the soil, right? So what is needed? What do they do? Basically, they are planting in what season? And if there is a special condition they would look for, they would definitely maintain it, that it should be there so that the seed can grow. Otherwise, their seed will be wasted. The crop will be wasted. Yes. Who is going to answer? Yes, Saad.
Sir, water is needed, and B and D have moist cotton wool.
First of all, very good. Water is needed. That's very true. It's a moist cotton. And first of all, we remember which part of the seed is going to absorb the water. I just want to integrate that information that we talked about seed germination. There's a particular part. Somebody would like to recall that name? Remember that part? A small hole where fusion of pollen, nuclei of the pollen is passing. Yes, Yusra?
Micropile.
Micropile. Yes, micropile is the small hole. There's this still, you know, the small growth. If you look at the beans, red beans, you might see on one side, the curved path, you might see a grain looking appearance, you know, grain appearance in one side of the seed. There is a micropile that is absorbing seed water. The water will get inside the seed because the rest of the part is covered with which covering? The covering of the seed that will protect the cotyledon is called? You remember the name of that part, the covering of the seed? Testa. Remember, testa is not porous. It's preventing water, because if water is getting in, so the seed will start germinating, right? So testa is protecting the seed, right? So the moisture cannot get in through this micropile until you have enough water. Okay, yes, Saad, you rightly mentioned. So what else is different in B and D? Now you can compare the whole things, you know, the whole experiment. they are giving you this B and D are germinating, the rest are not germinating. So first of all, which Saad is already picked on, is the moist contents. So the moisture is important. Water is important for the seed, because you remember in the seed cotyledon, the enzymes, the protein, starch, everything is dormant. Water should get in to activate the enzymes. Okay, what else is needed? Now compare the whole setup, all tubes. What else is needed? Look at this. Immediately you could compare temperature. What is, I mean, uncommon, which is very unique about this tube? What do you think? Temperature. This is not growing. Do you think in -4 it will grow? Do you think? So
No,
moisture
sir.
content is, yes, Yusra, go ahead.
No, I'm saying that at that temperature won't be able to grow since the
Yeah, so
photosynthesis will
yeah,
not be able
in single look, you could easily see what is different, -4. So of course, temperature is needed. So, and look at this boiled water. How much is the temperature? And oil is also on the top of it, so that it does not evaporate and doesn't cool down. And you have boiled water. If you put seed in boiled water, what will happen to the seed? So how boiling water will affect the seed? The seed will be damaged. Cotyledon starch will be just coming out of the seed, and it will be part of the water. And this extreme water temperature will damage the seed. And enzymes also. You remember the enzyme which are there in the cotyledon. Enzymes has got. Do you think that enzyme can work on 100 degrees Celsius boiling water? It is around 80, 90, not necessarily 100. If you pour it here, if you maintain it boiling, of course, boiling water will damage the enzyme. So seed is not going to grow. So temperature is important in two ways. First of all, extremely low temperature is not going to help the germination of seed. And second thing, extremely high temperature is not going to help the seed. Okay. And now look what is different in other tube. What is so common in B and D? So what is common here? Seed and moist cotton. Seed and moist, this is common, okay? So what is uncommon? Dry cotton. So there's why you may, it can make you think that moisture is needed, okay? And look at the curve. How does it help, you know? What does it give you extra information? It's still growing, although it's covered with black card. What does it mean? Yes, anybody would like to give? So you already mentioned that it's deprived of light. Black card mean light cannot pass through, okay? So it's a completely and dark, but the seed is growing, even if it is in the dark. What does it tell you? What will you conclude from this experiment here now? Yes, Beta Isra?
So I think that these seeds are grown in winter conditions because they do not require a lot of light.
Yeah, here, so you could say that light is not required initially for seed germination. As you remember, cotyledon, we mentioned about the cotyledon role. So it can give this energy and food for the seed germination. But when the shoot is coming out, definitely this plant is not going to survive. The shoot definitely is going to die if you keep maintain. But for germination, light is not needed, okay? For germination, as I mentioned, that you could use your common sense that when we are planting seeds, we are putting in soil, right? It is covered with dust, with earth, with the clay or sand. It's covered, right? It's inside the ground. It's not on the upper surface. So what does it mean? That sunlight is not necessary at that time. The sunlight that we need is maybe for the temperature, but this is showing another extra information: temperature, light, and moisture content. Quality of the seed was the same. Here the quality is somehow damaged because you have boiling water, right? So so what is your conclusion here? Moisture is needed, proper temperature is needed, okay? And the quality of the seed. What about B? Part B? they are giving another scenario using the same information, same experiment. They are saying that in the tropical rainforest, the trees often grow very closely together. They are thick forests because there are a lot of rains. You might see thick forests in Africa, in Western countries, or like in UK and US and Canada, some part of Canada. You might see rainfall is so much, and you could see thick garden. But in tropical also, we have a lot of rain formation. Rainfall is more. That's why, which reduces the amount of light reaching the forest floor. So one thing they are saying that if the forests are thicker, so one thing which they are saying, the light may not reach the floor. Okay, so they are giving you a very special challenge here. There is no light, or maybe very little light is there. So the seed of many species of the rainforest tree will not germinate until they get plenty of light. So what they are saying, that even if the seeds will fall on the ground, everything is fine, seed is there, but they are not receiving proper light, or although light is not needed, okay, but in the rainforest, this plant will not grow. Germination initial will take place, but they will not survive. The plant will die because there is no plenty light. So suggest why this is an advantage to the seedlings. What do you think? Why this will be an advantage to the seedling? Do you think is it an advantage or disadvantage? Who is going to answer this? Yes. So what will happen? What do you think? What will happen to, you can just compare with this experiment, right? Here it was light was needed, and temperature is somehow needed. Light here for the germination, as I mentioned, is not needed. But what do they mean here? That if even seed will germinate, this is not going to grow into a plant, because they don't it doesn't have enough light to grow. Initially the cotyledon will give some energy, and the shoot will grow, but it finally will die. It's not going to become a grown-up plant. And they say, well, how this is an advantage for the seedlings. So what do you think? So if the seed will grow, these seeds are not growing, one thing, right? And even the seedling will not, because even if these seed will germinate, the advantage is that they are not growing in less light, because even if the seed is germinating, the new shoots that are coming will die very soon, because ultimately it will need sunlight for photosynthesis. So photosynthesis cannot take place. So this is one of the advantages, that they should not grow until there's a plenty of light, because if they grow, they are going to die. So they're not going to survive, because they don't have enough light for the photosynthesis. And you might have seen it in the thick forest. If you go through the trails in the thick forest or in the bike area, you would see a lot of dead plants, small shooting plants, branches, yellow and dead. So this they cannot survive. So in a separate experiment, the student used seeds of rainforest trees. State the tube in the diagram above, in which the result would differ from those obtained for mustard seed. So what do you think? How you can correlate this scenario, okay, with your experiment that you have done it? And these are mustard seeds. They are saying if you take similar, which tube you are picking, that will closely resemble the rainforest, and what will be your analysis? What do you think? What will be your conclusion? They are now using, instead of mustard, you have rainforest seeds here, all rainforest seeds. So state the tube in the diagram here in all 1, 2, 3, 4, 5. Okay, in these 5 tubes, in which the result would differ from those he obtained from mustard seed. And how would be the result will be differ? So what? Okay, so which tube is deprived of light here? Which one? Yes, Yusra?
Tube B.
Tube D is deprived of light. And what happened to this these seeds in this experiment, in the mustard seed? What happened to this seed? Was it germinating or not germinating?
It
So
is
we mentioned
germinating.
that after 3 days, seeds in tube B and D were germinated. They had germinated here in B and D, right? So they have given you these information. Now it's fine. Now remove these mustard seeds, remove it, and take seeds of the rainforest trees, put it here. Remove these mustard seeds. So how would you think the result would be different here now? They have mentioned that these seeds cannot grow until there's enough light. This is a built-in quality in this seed. Allah subhanahu wa ta'ala has protected the generation of these plants. That if you remove these mustard seeds and you put the rainforest trees seeds here, they will not grow. They will not grow because there's no enough light. Mustard seed was growing, right? That's why it had germinated. The seed was germinated here. But if you exchange this, you replace these seeds with the rainforest seeds, they will not grow, right? So your observation is now different. You would see that only, you would say that's maybe another, if you will put in all of them, so you could say B had germination, not D. D will not be germinating because B has got more light for rainforest. This is one of the quality of the rainforest, that they are germinating. Okay, so let's see. What about the? Did we mention which part? One second. Yeah, so we mentioned part 2 here. Yeah, okay. So let's go to question 4. Okay, who is going to read this question? Yes, who is going to read this question, please, quickly? Yes, Yusra.
Question 4. The diagram below shows a banana plant producing suckers.
Okay.
L. Name the type of reproduction that is shown in the diagram.
So they are saying, you could see this is the whole banana growing plant. This is another plant, and there are small plants that are growing from the same plant. So they are saying now, name the type of this reproduction. You could see the plants are growing, right? If you remove this plant with little root and you put it somewhere else here in the soil, it will grow. Yes, Yusra. You would like to answer?
Yes, asexual reproduction.
This is asexual reproduction. Very good. Yes, good, Nabil. Good, Yusra. Very good. There's no pollination, no fertilization, no transfer of gametes, and just growing from the same. So this plant is having exactly it's a ditto copy of this plant, exactly the same. And as you rightly mentioned before, mitosis is taking place. Very good. Okay. Describe 2 advantages to growers of banana. Those farmers who are using banana plants by using this type of reproduction to propagate their plants. So just to, I think you can easily remember what will happen. Okay, I pick up this one, this sucker, the smaller shooting or smaller young plant, and I plant it here. So what is the advantage now? What do you think? What are the advantages to have such a crop or such garden of bananas? Any 2? Yes. So first of all, you think about, it will be very easy. You just cut it and you plant it here and it will grow very quickly. First of all, you can, in a very short time, you can have a garden of banana very easily. Now think about seeds. If you just put a seed here, you plant seed here, it might take time. Pollination will take place, and then seed formation, you have to grow the seed, it will grow into a plant, and finally another plant. So one of the advantages is that you are exactly, if this plant is of good quality, you are having the same properties in this small plant as well, right? Because you're taking the same part of the plant and planting somewhere else. So if this plant is good quality, the farmer will be happy with that. Oh, it all are the same quality. All banana are sweet. Not one orange is very sweet, another is sour, and some are really very bitter in taste. Why? Because they are generated from the seeds, right? So seeds, they have variety. It doesn't have variety now. So this is good thing, that if the quality is good, definitely farmers or gardener will be happy that they are having a good garden, right? And also very quick. Don't you think it will be very quicker? These two advantages are very quick. Okay, is it easy? Is it clear? You have any question? I hope it's clear. Okay, so think about now, banana plants can be killed by fungal disease such as black Sagataka and Panama disease. These are, I think, some locality in Europe, Fiji land. These are the some disease or fungal disease of plants. Explain why a population of banana produced by this method, shown in the diagram, could all be wiped out by the same disease. I think this I would like you to answer, please. Yes, Yusra.
Since they are being produced by the asexual reproduction, their DNA has been replicated, which is like having the same similar properties from their parent plant. So if the parent plant is having the disease, the furthermore plants will be having the same disease.
Wonderful, excellent. There's no diversity. There is no resistance. There is no genetic variation. They are all identical. If one plant is affected by any disease, is going to ruin the whole crop, the whole garden, because these plants are not different from the parents, right? You have grown it, cutting this part of the plant, you plant it here, and then you cut another part and plant it there. You have a whole garden having same genetic makeup, genetic properties, genetic qualities, okay? And that's why you might have seen plants that we are growing in back in our country from seeds. Some plants will die. You might see in wheat, I think this is very common, a black powder, a part of the seeds are getting disease. The bunch will get a disease, that is a fungal disease. So not necessarily the whole crop is damaged by that disease, because they are grown up from the seeds. So these plants are not genetically identical. Okay, of course the species is the same. You might see little difference in quality, the look is the same, you might see taste is the same. It doesn't mean that one mango is totally different from another. Yeah, taste might be different, but yes, due to genetic diversity or genetic makeup is different. That's why these plants grown from seeds will have more resistance against disease. These bananas cannot. Why? Because they are grown from the same. Okay, so from the same plant, one parent is contributing asexually, right? It's not hermaphrodite as well, because there is no pollination. Okay, so that's all about, I think, now I have to conclude, although this is one question, but it's really long question. We need to have a very good discussion on this, because this is a very special kind of primrose flower. I would say that please type in Google and see primrose flower. How they are different? Why they are called pin eye? Why they are called thumb eye? There's a pin kind. When you look at the flower, just a pin will be showing in the center of the flower, and here is like a bunch of anthers. So you read it, inshallah, and we'll cover it in the next session. Okay, we'll conclude it. And if you have any question, anything you would like, I can give you just 1 or 2 minutes. Otherwise, we'll conclude. Is it fine? Okay, then, inshallah. See you next time. Jazakallah khairan. Assalamu alaikum wa rahmatullahi wa barakatuh.
Walaikum assalam. Allah Hafiz.
Allah Hafiz.